Good day, everyone. Our topic for today is all about gene functions. Here are the reporters for today's discussion. And for our objectives, at the end of this video, learners are expected to describe the nature of genetic code, understand the information transferred from DNA to mRNA to protein by transcription and translation, and evaluate the importance of gene functions into our body and other living organisms. And here's our topic outline in gene functions. First, we have the genetic code. Second, the transcription process in prokaryotic and eukaryotic. Third, the RNA processing in eukaryotic cells. Fourth, the translation, which has three subtopics, which is the translation process and the collinearity of DNA and protein. Second, the protein structure and functions. Third, the regulation of gene expression.
Before we go to genetic code, let us first introduce DNA. The DNA acts as a genetic material that encodes all the information required to encode a protein. A specific region of DNA encodes the genetic information is called a gene. There can be several genes in a genome. It is usually the process of transcription. The genetic information in the DNA flows in RNA, also known as messenger RNA or mRNA. The information contained in mRNA is further used for translation to make proteins. This flow of information from DNA, RNA, and proteins is also known as the central dogma of molecular biology. So now let's talk about genetic code. Genetic code is defined as as a sequence of three nucleotides in RNA that determines the sequence of amino acids for the synthesis of proteins. In other words, the genetic code is defined as the set of rules by which information encoded in DNA or RNA sequences is translated into proteins by the living cells. Now let's get to know Marshall Nirenberg, the father of genetic code. Marshall Nirenberg, a biologist who deciphered the genetic code of life, earning the Nobel Prize for his achievement. In 1959, Nirenberg began his investigation in the relationship between the deoxyribonucleic acid or DNA, ribonucleic acid or RNA, and the production of proteins. In solving the genetic code, Dr. Nirenberg established the rules by which the genetic information in DNA is translated into proteins and working parts of living cells. So here is the genetic code table. The genetic code table is used for translating a genetic code into sequence of amino acids. Properties of genetic code. Codes are First is the codes are triplet. Each genetic code is triplet form of three nucleotide bases that code for a particular amino acid. There are 64 codons that code for 20 amino acids, which means that there exists more than one code of single amino acid. Second property is codons are non-overlapping. The genetic codes are non-overlapping. The adjacent codons do not overlap. It means that the same letter is not used for two different codons. A single base cannot take part of the information of more than one codon while reading the sequence of bases from the starting of mRNA in blocks of 3. The third property is codon are universal. The genetic code in universal, which means the genetic code is universal, which means the same codons code for the same amino acid in all forms of life that exist today. Fourth property is codons are commaless. The genetic code is commaless, which means codons are read continuously. The deletion of single base in a commaless code alters the entire sequence of amino acids. Fifth property is codons are non-ambiguous. The genetic code is non-ambiguous and specific because each code codes for only one amino acid. The sixth property is codons are degenerate. Several codons code for the same amino acids except for tryptophan and methionine, which are coded by one codon each. This multiple system of coding for the same amino acids are known as degenerate coding system. It involves protection to the organism against many harmful mutations. Last property is codons have polarity. The genetic codes has a definite direction for the reading of information which is called polarity. If codon is read in the opposite direction, then it would specify another amino acid because of the alteration of base sequences in the code. The message in the mRNA is read to 5 to 3 direction, and thus the polarity of genetic code is from 5' to 3'. So next is the types of genetic code. First is sense codons. There are there are defined as those codons that code for amino acid. There are 61 codons in the genetic code that code for 21 amino acids. Second is signal codons. These are defined as those codons that code for amino acids that acts as signal during protein synthesis. There are four codons that acts as signal, namely AUG, A, UAA, UAG, and UGA. Here are types of signal codons. First is the start codons. These are those codons that starts the translation process. They are also known as the initiating codons as they initiate the protein synthesis process. Second is stop codons. These are those codons that provide a signal for the termination of polypeptide chain synthesis. They are also known as termination codons as they stop the formation of polypeptide chains. Lastly, exemptions to genetic code. The universality of genetic code conveys that similarity codons are assigned to identical amino acids, along with similar starts and stop signals in the genes of most microorganisms and plants. Out of the few exemptions that were discovered, most of them includes assigning one or two stop codons to an amino acid. Another exemption includes two codons, GUG and AUG. Both of their codes are amino acid methionine acts as starting codons, although GUG is meant for valine. This exception deviates one of the properties of the genetic code. Genetic codes are non-ambiguous. Thus, it confirmed that few codes often differ from the properties of non-ambiguous.
Transcription of prokaryotic and eukaryotic cells. So, have you ever had to transcribe something, or maybe someone left a message on your voicemail and you had to write it down on paper? As these examples show, transcription is a process in which information is rewritten. Transcription is something we do in our everyday lives, and it is also something our cells must do in a more specialized and narrowly defined way. In biology, transcription is the process in which a gene's DNA sequence is copied or transcribed to make an RNA molecule. Transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as a protein. Transcription uses one of its two exposed DNA strands as template. This strand is called the template strand. The RNA product is complementary to the template strand and is almost identical to the other DNA strand called the non-template or the coding strand. However, there is one important difference. In a newly made RNA, all of the T nucleotides are replaced with the U nucleotides. Proceed to prokaryotic transcription. It is the process in which messenger RNA transcripts of genetic material in prokaryotes are produced to be translated for the production of proteins. Prokaryotic transcription occurs in cytoplasm alongside translation. Prokaryotic transcription and translation can occur simultaneously. The main enzymes involved in transcription is the RNA polymerase, which uses single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the fifth to third direction, adding each nucleotide to the third end of the strand. RNA polymerases are large enzymes with multiple subunits, even in simple organisms like bacteria. Humans and other eukaryotes have three different kinds of RNA polymerase that specializes in transcribing certain classes of genes. So to begin transcribing a gene, RNA polymerase binds to the DNA of the gene at a region called the promoter. So basically, the promoter tells the polymerase where to sit down on the DNA and begin transcribing. To get a better sense of how a promoter works, let's look at an example from bacteria. A typical bacterial promoter contains two important DNA sequences, the negative 10 and negative 35 elements. RNA polymerase recognizes and binds directly to these sequences, and the sequences position the polymerase in the right spot to start transcribing a target gene, and they also make sure it's pointing in the right direction. Once RNA polymerase is in position at the promoter, the next step of transcription, which is elongation, can begin. Basically, elongation is the stage when the RNA strand gets longer thanks to the addition of the new nucleotides. During elongation, RNA polymerase walks along one strand of DNA known as the template strand in the third to fifth direction. For each nucleotide in the template, RNA polymerase adds a matching complementary RNA nucleotide to the third end of the RNA strand. The RNA transcript is nearly identical to the non-template or coding strand of DNA. However, RNA strands have the base uracil in place of thymine, as well as a slightly different sugar in the nucleotide. So as we can see in the PowerPoint presentation, each thymine of the coding strand is replaced with the uracil in the RNA transcript. So the last step is determination. So in a prokaryotic cell, by the time transcription ends, the transcript would already have been used to begin making copies of encoded protein because the process of transcription and translation can occur at the same time since both occur in the cytoplasm. For the next slide is determination in bacteria. So there are two major strategies found in bacteria, the row dependent and the row independent. So in row dependent termination, the RNA contains a binding site for a protein called row factor. So this row factor binds to the sequence and starts climbing up to the transcript towards RNA polymerase. And for the row independent termination, it depends on specific sequences in the DNA template strand. As the RNA polymerase approaches the end of the gene being transcribed, it hits a region rich in cytosine and guanine nucleotides. The RNA transcribed from this region falls back on itself and the complementary cytosine and guanine nucleotides bind together. The result is a stable hairpin that causes the polymerase to stall. So let's proceed to the eukaryotic transcription. So unlike prokaryotic RNA polymerase that can bind to a DNA template on its own, eukaryotes require several other proteins called transcription factors to first bind to the promoter region and then help recruit the appropriate polymerase. The completed assembly of transcription factors and RNA polymerase bind to the promoter, forming a transcription pre-initiation complex or PIC. So there are three eukaryotic RNA polymerases, which is the RNA polymerase 1, 2, and 3. The RNA polymerase 1 is located in the nucleolus, a specialized nuclear substructure in which ribosomal RNAs is transcribed, processed, and assembled into ribosomes. The next one is the RNA polymerase 2, which is located in the nucleus and synthesizes all protein coding nuclear pre-RNAs or messenger RNAs. Eukaryotic pre-mRNAs undergo extensive processing after transcription but before translation. So RNA polymerase II is responsible for transcribing the overwhelming majority of eukaryotic genes, including all protein coding genes. The next and the last one is the RNA polymerase III, which is also located in the nucleus. This polymerase transcribes a variety of structural RNAs that includes the 5S pre-RNAs, transfer-free RNAs, and the small nuclear RNAs. So for the eukaryotic initiation stage, it is where a generalized promoter of a gene transcribed by RNA polymerase II is shown. The transcription factors recognize the promoter. The RNA polymerase then binds and forms the transcription initiation complex. For the next slide is the promoters for humans or eukaryotes. In eukaryotes like humans, the main RNA polymerase in your cells does not attach directly to the promoters like bacterial RNA polymerase. Instead, helper proteins called basal or general transcription factors bind to the promoter first, helping the RNA polymerase in your cells to get foothold on the DNA. The data box, as you can see in your in the PowerPoint presentation, as the core promoter element is the binding site for a transcription factor known as the Tata binding protein or the TBP, which is itself a subunit of another transcription factor, which is the transcription factor 2D. After TF2D binds to the Tata box via TBP, five more transcription factors and RNA polymerase combine around the Tata box in a series of stages to form a pre-initiation complex. For the elongation stage, once the DNA strand is properly positioned in the metal ion active site of polymerase II, elongation begins. So the elongation phase of transcription is broken down into three stages, which is the promoter escape, promoter proximal posing, and productive elongation. So the first stage of elongation is the promoter escape, which involves the maturing the RNA polymerase II molecules so it is capable of staying in contact with the DNA template strand throughout productive elongation. During this stage, the polymerase breaks its contact with some of the promoter sequence elements and promoter bound factors while tightening its hold on the elongating RNA molecule. Once the growing RNA molecule forms a stable association with the transcription complex, the next stage of elongation, which is the promoter proximal posing, begins. So during the promoter proximal posing stage, the polymerase II enzyme poses in the fifth region of the transcription complex, and upon the appropriate signals, it progresses to productive elongation. So the next and last step is the productive elongation, which is the final stage in the elongation phase of transcription. During this stage, the polymerase II moves through the rest of the gene being transcribed. So the last step for the eukaryotic transcription is the termination. So termination follows elongation and is crucial step in the transcription process. Many consequences are possible if polymerase II never responds to termination signals. For example, failure to end elongation can lead to reduction in the expression of a downstream gene by interfering with the initiation complex of that gene. It could also obstruct the correct transmission of eukaryotic chromosomes. This leads to the dissociation of the complete transcript and the release of RNA polymerase from the template DNA. So the process differs for each of the three RNA polymerases. So that ends my report for transcription process of prokaryotic and eukaryotic cells.
And for our next topic, it is all about the RNA process in eukaryotic cells. So before dwelling into the process of RNA in eukaryotes, I want you to imagine that you run a bookmaking factory and you've just printed up all the pages of your favorite book. Now that you have the pages, is the book ready to go? Well, books usually have front and back covers, so you might want to put those on. Also, were there any blank or messed up pages made during printing? You should probably check for those and remove them before selling your books, or you might end up with some unhappy customers. So, the steps we just talked about are pretty similar to what happens to RNA transcripts in the cells of your body. In humans and other eukaryotes, a freshly made RNA transcript is not quite ready to go. Instead, it is called a pre-mRNA and has to go through some processing steps to become a mature messenger RNA or mRNA that can be translated also into protein. So these include the following steps. We have the addition of cap and tail molecules to the two ends of the transcript. So these play a protective role like books front and back covers. So removal of junk sequences, that is called the introns. So introns are sort of like blank or messed up pages made during the book's printing, for example. So which have to be removed in order for the book to be readable. No? So later on we will talk about this. So in this point we will take a closer look at the cap, tail, and splicing modifications that eukaryotic RNA transcripts receive, seeing how they carried out and why they are important for making sure we get the right protein from our RNA. But before that, we will have an overview no, of the pre-mRNA processing in eukaryotes. So as a quick review, gene expression happens a little bit differently in bacteria and eukaryotes such as humans. So we have here a simple diagram of bacteria and eukaryotes. So as you see, it's a little bit simpler in bacteria. You have DNA just floating around the cytosol, and so the transcription takes place. So you start with that DNA, that protein coding gene in the DNA, and from that you code the messenger RNA. And you see in that purple color rather, right over here, and then that messenger RNA can be involved with the ribosome and that translation process to actually produce the polypeptide and to produce the protein. But on the other hand, these eukaryotic cells, we're going to get into a little bit more depth now in this discussion. So that transcription, the DNA to mRNA, that happens inside the nucleus. So there's essentially two steps here, no? You go from DNA to what we call pre-mRNA, and it needs to be processed to turned into what we call mRNA. So which can leave the nucleus to be translated into protein. So now that we have the overview, so let's dig deeper into this and understand what type of processing might actually going on. So again, these are the steps or key points in this topic. So we have a 5 cup is added to the beginning of the RNA transcript, and 3A poly A tail, 3 poly A tail rather, is added to the end. So in splicing, some sections of RNA transcripts that we call introns are removed, and the remaining sections or exons are stuck back together. So some genes can be alternatively spliced and leading to the production of different mature mRNA molecules from same initial transcript. So with these key points, let's talk about this one-on-one. No? So 5 cup and poly A tail. So both ends of a pre-mRNA are modified by the addition of chemical groups. So the group at the beginning 5 end is called a cap, while the group at the end or kaning 3 end is called a tail. So both the cap and the tail protect protect the transcript and help it get exported from the nucleus and translated on the ribosomes or those protein making machines. This will be discussed now after this, and that is found in the cytosol. So the 5 cap is added to the first nucleotide in the transcript during transcription. So the cap is a modified guanine or G nucleotide, and it protects the transcript from being broken down. And it also helps the ribosome attach to the mRNA and start reading to make a protein. So how is this poly A tail added? So the 3 end rather of the RNA forms in the kind of bizarre way when a sequence called polydenylation signal shows up in the RNA molecule during transcription. An enzyme chops the RNA into at that site. Another enzyme adds about like 100 to 200 adenine A nucleotides to cut the end, forming the poly A tail. So the tail makes the transcript more stable and helps it get exported from the nucleus to the cytosol. So that is what happened in this stage. No, 5 cup or is added in the beginning, and 3 end is added in the tail. So now let's proceed to the RNA splicing. So the third big RNA processing event that happens in your cell is this RNA splicing. So in RNA splicing, specific parts of the pre-mRNA called introns are recognized and removed by a protein and RNA complex called the spliceosome. So introns can be viewed as junk, no sequence that must be cut out so the good parts or version of the RNA molecule can be assembled. So unsa rin good parts? What are these good parts? So the pieces of the RNA that are not chopped are called exons. Exons. So the exons are pasted together by the spliceosome to make the final and mature mRNA that is shipped out of the nucleus. So that is what happened in this diagram here shown. So a key point here is that it's only the exons of a gene that encode a protein. Not only do the introns not carry information to build a protein, they actually have to be removed, no, in order for the mRNA to encode a protein with the right sequence. So if the spliceosome fails to remove an intron, and mRNA with extra junk in it will be made. So, and a wrong protein will get produced during the translation. So, wrong if dili ma-remove ang introns. And another ways another process rather is the alternative splicing. So why we need to splice? So we don't know for sure why splicing exists, and in some ways it seems like a wasteful system. But however, splicing does not allow for a process called alternative splicing, in which more than one mRNA can be made from the same gene. So through alternative splicing, we can sneakily encode more different proteins than we have in our genes or in our DNA. So in alternative splicing, one pre-mRNA may be spliced in either of the two, or sometimes many more than two in different ways. For example, in diagram below, the same pre-mRNA can be spliced in three different ways depending on which exons are kept. So this results in three different mature mRNAs, each of which translate into protein with different structure. So this is why. So this exists. One possibility is that splicing in general permits alternative splicing, which appears to be important for eukaryotes, no, by allowing them to make more different protein from a single gene. Also, introns, sometimes called regulatory sequence that control of the expression of a gene. Sometimes small RNAs that regulate gene expression come from introns that are also spliced out. Also, the existence of exons and introns may make it easier for the new genes and allele to form through mutation events that mix and match gene segments, which could have evolutionary advantage. So we have here, write yourself the splice message, no, to apply the RNA process in eukaryotes. So your mission your mission in this is decode the following top secret message. First, remove the junk or the letters colored in purple and underlined. The second, put the remaining letters into groups of 3, starting at the beginning. So we have here kanang set of letters. No? If you remove the purple sequence, you should get this series of letters. So this one, you should get these series of letters. So the dog run, but if you group the remaining letters into 3, you should get this message. So this is the secret message. This is the dog ran and ate the hat. So the process you just went through is basically what your cells must do when they express a gene. So as we discussed earlier, most eukaryotic pre-mRNAs contain junk sequences called introns, which are like the purple letters in this message. So these sequences must be removed. And the meaningful sequences or exons equivalent to the maroon letters in the message above must be stuck together to make the mature mRNA. So during the translation, the mRNA sequence is read in groups of 3 nucleotides. So each 3-letter word corresponds to an amino acid that's added to a polypeptide or the protein subunit. So if an RNA hasn't wala siya na splice, it will contain extra nucleotides that it shouldn't, leading to an incorrect protein message. So something similar happens if we try to decode the message below without removing the purple letters. So if we try to decode the message earlier, so this is what we get. Just as removing the purple letters from the sentence is the key ending up with the right message. So splicing is the key to ensuring that the mRNA carries the right information and directs production of the correct polypeptide. So another example of the pre-mRNA sequence with exons in bold maroon letters and one intron in bold purple letters. So we have here. So if the pre-mRNA is correctly spliced, the nucleotides of the mature mRNA are read in a groups of 3, follows producing the polypeptide shown below. So we have here, if we group this bunch of letters above into 3, so we got this here, and we also remove these purple letters now because these are introns. So if the introns is not removed from the pre-mRNA, the resulting abnormal mRNA contains extra nucleotides. So when its nucleotides are read in groups of 3, now as shown here, a polypeptide with an incorrect amino acid is produced. So this is incorrect because the introns rather are not removed, and yeah, and the what we call here, the message will be incorrect. So all of the above relationships between group of nucleotides and amino acids they encode are based on the determined using the genetic code table. So that's it for the RNA process in eukaryotic cells. I hope you learned something.
Since we already established that a genetic code stores information in the form of triplet nucleotides in DNA, and that this information is initially expressed through the process of transcription into a messenger RNA that is complementary to one strand of the DNA helix. Now we will proceed to the translation process that involves decoding a messenger RNA and using its information to build a polypeptide or chain of amino acids. Translation can be divided into 3 steps. First is the initiation. In order for translation to start, there are few key ingredients that are needed. These include a ribosome, an mRNA, an initiator mRNA. So during the translation, these pieces must come together in just the right way. Together they form the initiation complex, the molecular setup needed to start making a new protein. So the question here is, does that just happen by itself? No, not exactly. Initiation calls for some helper molecules, as well as an energy source. Initiation depends on specialized proteins, helpers called initiation factors. Their job is to help a ribosome subunits, tRNA and mRNA, find each other in an orderly and predictable way. Now let's look into what's inside a eukaryotic cell during translation. So the tRNA carrying methionine attaches to the small ribosomal subunit. Together, they bind to the 5' end of the mRNA by recognizing the 5' GTP cap. Then they walk along the mRNA in the 3' direction, stopping when they reach the start codon, which is the AUG. On the other hand, in bacteria, the situation is a little different. Here, the small ribosomal subunit does not start at the 5' end of the mRNA and travel toward the 3' end. Instead, it attaches directly to certain sequences in the mRNA. These Shine-Dalgarno sequences come just before start codon and point them out to the ribosome. So why use Shine-Dalgarno sequences? So bacterial genes are often transcribed in groups called operons that will be discussed later. So one bacterial mRNA can contain the coding sequences for several genes. A Shine-Dalgarno sequence marks the start of each coding sequence, letting the ribosome find the right start codon of each gene. Next step is elongation. Elongation is when the polypeptide chain gets longer. But how does the chain actually grow? So to find out, let's look at the first round of elongation. The methionine-carrying tRNA starts out in the middle slot of the ribosome, called the P site. Next to it, a fresh codon is exposed in another slot, called the A site. The A site will be the landing site for the next tRNA, one whose anticodon is a perfect or complementary match for the exposed codon. Once the matching tRNA has landed in the A site, it's time for the action. That is the formation of the peptide bond that connects one amino acid to another. This step transfers the methionine from the first tRNA onto the amino acid of the second tRNA in the A site. Once the peptide bond is formed, the mRNA is pulled onward through the ribosome by exactly one codon. This shift allows the first empty tRNA to drift out via the E site or the exit, and it also exposes a new codon in the A site, so the whole cycle can repeat. So last step is the termination. Termination happens when a stop codon in the mRNA enters the A site. These stop codons referring is the UAA, UAG, and UGA. So stop codons are recognized by proteins called release factors, which fit neatly into the P site. Release factors mess with the enzyme that normally forms peptide bonds. they make it add a water molecule to the last amino acid of the chain. This reaction separates the chain from the tRNA, and the newly made protein is released. Next to be discussed is all about the collinearity of DNA and protein. So what does it mean? So it simply means that the length of a DNA sequence in a gene is proportional to the length of polypeptide encoded by the gene. Meaning, longer genes encode bigger proteins. Another thing is the protein structure and function. There are four. First is the primary structure, which is defined as the linear amino acid sequence of a protein polypeptide chain. In fact, the term protein sequence is often used interchangeably with primary structure. Next is the secondary structure, which is defined as the local spatial conformation of the polypeptide backbone excluding the side chains. Third is the tertiary structure, which refers to the three-dimensional arrangement of all the atoms that constitute a protein molecule. It relates the precise spatial coordination of secondary structure elements and the location of all functional groups of a single polypeptide chain. And lastly, the quaternary structure, which is of many structured proteins are oligomers consisting of more than one polypeptide chain. So the precise spatial arrangement of the subunits within a protein, which is referred as the quaternary structure. Among all, another concept we need to know is how is gene expression being regulated. So there are various forms of gene regulation, that is, mechanisms for controlling which genes get expressed and what levels. However, a lot of gene regulation occurs at the level of transcription. So bacteria have specific regulatory molecules that control whether a particular gene will be transcribed into mRNA. Often, these molecules act by binding to DNA near the gene and helping or blocking the transcription enzyme RNA polymerase. So let's take a closer look at how genes are regulated in bacteria. So in bacteria, genes are often found in operons. Related genes are often found in a cluster on the chromosome, where they are transcribed from one promoter as a single unit. Such as a cluster of genes under control of a single promoter is known as an operon. Operons are common in bacteria, but they're rare in eukaryotes such as humans. In general, an operon will contain genes that function in the same process. For instance, a well-studied operon called the lac operon contains genes that encode proteins involved in uptake and metabolism of a particular sugar, lactose. So operons allows the cell to efficiently express sets of genes whose products are needed at the same time. So operons aren't just made up for coding sequences of genes. Instead, they also contain regulatory DNA sequences that control transcription of the operon. Typically, these sequences are binding sites for regulatory proteins, which control how much the operon is transcribed. The promoter, or site where RNA polymerase binds, is one example of a regulatory DNA sequence. So most operons have other regulatory DNA sequences in addition to the promoter. So these sequences are binding sites for regulatory proteins that turn expression of the operon up or down. So where do the regulatory proteins come from? Like any other protein produced in an organism, they are encoded by genes in the bacterium genome. So the genes that encode regulatory proteins are sometimes called regulatory genes. So many regulatory proteins can themselves be turned on or off by specific small molecules. So the small molecule binds to the protein, changing its shape and altering its ability to bind DNA. For instance, an activator may only become active or be able to bind DNA when it's attached to a certain small molecule. Additionally, operons may be inducible or repressible. So some operons are usually off but can be turned on by a small molecule. So the molecule is called an inducer and the operon is said to be inducible. For example, so the LAC operon is an inducible operon that encodes enzymes for metabolism of sugar, lactose, and it turns on only when the sugar lactose is present. And the inducer in this case is allolactose, which is a modified form of lactose. On the other hand, other operons are usually on but can be turned off by a small molecule. So the molecule is called a co-repressor and the operon is said to be repressible. For example, the TRP operon is a repressible operon that encodes enzymes for synthesis of the amino acid tryptophan. This operon is expressed by default but can be repressed when high levels of the amino acid tryptophan are present. The co-repressor in this case is tryptophan. So these examples illustrate an important point that gene regulation allows bacteria to respond to changes in their environment by altering gene expression, and thus changing the set of protein present in the cell. On the other hand, let's now look into the gene regulation in a eukaryotic cell. The expression involves many steps, unlike in the prokaryotic, and almost all of them can be regulated. So different genes are regulated at different points, and it's not uncommon for a gene to be regulated at multiple steps. So first step is chromatin accessibility. So the structure of chromatin, DNA, and its organizing proteins can be regulated. More open or relaxed chromatin makes a gene more available for transcription. Next is transcription. Transcription is a key regulatory point for many genes. Set of transcription factor proteins bind to specific DNA sequences in or near a gene and promote or repress its transcription into an RNA. Third is the RNA processing. So splicing, capping, and addition of a poly A tail for an RNA molecule can be regulated, and so can exit from the nucleus. So different mRNAs may be made from the same pre-mRNA by alternative splicing. Next is the RNA stability. So the lifetime of an mRNA molecule in the cytosol affects how many proteins can be made from it. So small regulatory RNAs called miRNAs can bind to target mRNAs and cause them to be chopped up. Next is the translation, which translation of an mRNA may be increased or inhibited by regulators. For instance, miRNAs sometimes block translation of their targets mRNAs rather than causing them to be chopped up. And lastly, the protein activity. So proteins can undergo a variety of modifications, such as being chopped up or tagged with chemical groups. These modifications can be regulated and may affect the activity or behavior of protein. Although all stages of gene expression in a eukaryotic cell can be regulated, the main control point for many genes is transcription. Later stages of regulation often refine the gene expression patterns that are roughed out during transcription. And that ends our discussion. Thank you.